So this is a video about how to use a Python package called smallrnaseq to analyze the short non-coding RNAs present in short read sequence data, that is usually FASTQ files. And this video will concentrate mostly on how to use the command line interface, which is designed to be flexible but relatively straightforward to use. So if you're watching this, then it means you probably already know about the experimental methods to build the libraries and send off your sample for sequencing. And when that comes back, you'll have a set of FASTQ files. And usually this workflow here shows a somewhat simplified outline of the computational methods needed to get counts of these short non-coding RNAs in your sample, which could be microRNAs, SNO-RNAs or pRNAs, or there's a host of others. And usually the first step involved is to trim the adapters. That's the removal of usually the 3' adapter. And the next is to collapse the reads. And what this means is to find all the unique reads and remove duplication by keeping only one instance of each unique read and recording the copy number. And then this results in a much shorter file that is passed to a short read alignment algorithm like bowtie, and collapsing the reads makes this process of alignment much faster. And when that's done, the alignment will produce sorry, I should say the alignment is done to some known gene annotations that you download from a gene database like Ensemble, RNA Central, or maybe Mirbase, or to a reference genome. And once the reads are aligned, you can count the reads present in known genes or predict new genes, for example, the prediction of novel microRNAs. And often then the counts will be post-processed by normalizing them in order to do, say, a differential expression analysis. So the rest of this video is an overview of how to use this package, or smallrnaseq, to perform this workflow. And anyone who's familiar with the RNA-seq analysis in general can see that it's essentially the same methodology, with the main difference for small RNAs being the length of the reads, which are usually below 50 nucleotides. And that really is important later on when you come to do the alignment, as we'll discuss. So this tutorial is mainly meant for non-bioinformaticians who aren't that familiar with this kind of analysis, or where to download the gene lists or the gene annotations that you need to actually count what's in your samples. So there's plenty of resources for RNA gene annotations, and this is one of them, RNA Central. And it works pretty much as you'd expect. It allows you to do a search by, say, species name or RNA type. In this case, I'm looking for RNA genes for cow. So I do a search, and it brings me up a results list with the different kinds of RNAs in the menu here on the left. And so intuitively you can see if you just want to find microRNAs, you click on here for RNAs, for example, ribosomal RNAs. It gives me 1000 sequences, and I can download them then by selecting here and clicking on FASTA, and then this query will be submitted and the download link will appear here. When that's finished, sorry, that sometimes takes a couple of minutes. In fact, so I downloaded the file of cow ribosomal RNAs using this database, and I've also fetched a list of transfer RNAs, annotated transfer RNA genes for cow using this one, the Genomic Transfer RNA database, which works in a similar way. And if we search for cow, it'll give us results for two different gene assemblies. So I pick this UMD, and then it allows me to download the FASTA sequences, which actually opens directly in the web page here, and you can save as a text file. So there's plenty of other resources available, and later I'll talk a bit about downloading reference genomes using Ensemble, this page, or the UCSC browser. So for now, I've already downloaded the RNA gene files from these databases. These are transfer RNAs for cows from the genomic transfer RNA database, and the gene annotations of ribosomal RNA for cows. So these two FASTA files are placed in a folder along with the sample I want to run on, which is a FASTQ file. It's about 1 gigabyte in size, and that's this is from a serum sample in cow. And in order to run the program, it's simply from the command line we run smallrnaseq, and if I run that on its own, it'll generate this output. So the program is based around really using a configuration file, which is just a text file. And because there was none in the folder called default.conf, it's created that one there. And when you run the program then properly, you're going to provide this as an input along with -r to run the workflow. So if we open this configuration file in the text editor, you see that the layout is fairly straightforward. It's made up of sections. The base section is for all the general settings. These other sections are for different things we don't have to worry about for the moment. This is for part of the settings for novel microRNA prediction. This is for changing aligner parameters. This is for differential expression. But we just want to concentrate for this simple example on a couple of the options. So the first thing we want to do here is to just tell it what file we want to run. So that's tell it the file name. It's a single file name. We can provide more than one. So in this case, the name of our file is this one. We just paste it in, and then we also want to tell it which libraries we want to map to. That's these files here. But before we can do that, we want to make indexes for each of these, so that with Bowtie, so Bowtie will be able to do the alignment, and that's simply done. It can be done directly using Bowtie from the command line, but to make it simple, we have an option here. If I say smallrnaseq -h, it'll show us the options, and one of the options is -b, which says build an index for a FASTA file. So if I simply say run smallrnaseq -b and give it the name of one of the files, in this case say the ribosomal RNA FASTA file, and that creates a Bowtie index by default in a folder called indexes. And you can change this and then tell the program where your indexes are using this option here, but for now we'll just leave it at this. And I've already run it for the other file here, so the indexes are there, and all we simply have to do now, the last thing we have to do within the configuration file then is to tell it which libraries we want to map to, which is just a comma separated list of the names without file extensions, and we save the file. And then to run this now, these are the only options we need to add. You can see here the default aligner is Bowtie. We're going to leave it at that. Bowtie is installed on this system, and then to run it we simply say smallrnaseq -c default.conf, the configuration file, and then -r is to run. And the first thing it does is it spits out the current options we're using. It's found one input file. It's going to map to these libraries, but the first thing it does is it has to collapse the reads, and that can be possibly the longest part of the process. So for say a read file that has maybe 10 million reads, it might take 30 seconds up to a minute to do each file. So when that's done, it'll output how many reads have been collapsed and how many unique reads it's found. Then it'll be able to use this file as an input to Bowtie. This is the Bowtie command for the first file, and then the remainder are passed on to the next file, and so on. And then at the end, it'll report that it saved the results to a CSV file, comma separated file, and also it'll produce a table here of the fraction of each annotation, each library that it's mapped, and the remainder are the unmapped. These are pretty rough estimates, and they'll be very dependent on the parameters you've used for the aligner, which are this. So these are the default parameters. They can be changed, and they can be changed per index or per library that you map. So in the results folder here, it'll produce a couple of plots, and one of them is this. This is just a visualization of this table showing the fractions of each library that's been mapped. So this process acts to give you a good rough estimate of what's in your sample, essentially. And it's going to be, like I said, dependent on your aligner settings. If you don't understand these settings in Bowtie, you can look up the documentation or a tutorial on YouTube, but you can see here, for example, that this option simply means allow one mismatch. So obviously if you allow 0 mismatches, you're going to be able to map less reads, and these results will be slightly different. So the last example was a simple example of a single FASTQ file and mapping to a couple of annotated library indexes. And the only other thing to mention about that is that it produced an rna-count.csv file, which we can open in a spreadsheet to have a quick look at. And that just shows you what's inside, which is the number of reads mapped to each gene in the annotation. And there will be a column. In this case, there's only one sample, column for each sample. There's a normalized column. There's a column for total reads and mean normalized reads. Counts. And these are all just transfer RNA genes. In fact, this may not be particularly useful to you at this in this example, because you might only be interested in the fractions of types of RNA in your sample, and that might be enough for you. But it becomes useful later on in the next example that we're going to look at, which is for microRNA counting. So we go back out to another folder, this one here, we want to run the microRNA example. And that's going to be run on, I'll just delete these, that's going to be run on these 3 test files here, which are also taken from cow samples. So we're going to use the same, some of the same files that we used previously. But in this case we want to also count microRNAs explicitly. So we're going to use this configuration file here, which you open again in a text editor. And if you look at this, I've already entered the settings. So the difference in this case is we don't just specify a single file. We're going to just tell it to use all the files in this path. So that's the path option here. All the files in this folder, test files. And I've used a different index path for the Bowtie indexes. That's because the reason for that is I've already downloaded a reference genome for cow, which I'm going to need later on when we want to do novel microRNA prediction. So in order to do that, it has to map to the reference genome, and I've put the index for that in this folder here. So we use that as the index path. We're also going to, because we're doing microRNA prediction, we have to set this option here to be 1, and then we have to tell it the species. So the you don't have to download individually separately files from Mirbase or anywhere else for microRNA annotations. It's built in. Once you tell it the species, it'll generate the mature and precursor sequences. The only other thing here is that we still want to map to some libraries, and if you enter these as we did in the last example, it'll map to these, it'll align to these first, and it'll map the remainder of the reads to the microRNAs. So this allows you maybe to exclude noise basically from your samples if they contain a lot of transfer RNAs or RNA fragments that you don't want to include in microRNA counting, and particularly for novel microRNA prediction later, to reduce the amount of false positives. And the other samples, the other options in the novel section here are to set a threshold for the score cutoff when it's scoring precursors. But if you don't have to worry about them too much, except to say that reducing the score cutoff will potentially increase the number of false positives, but also give you more results. In fact, I've already tried to run this and had a problem with memory, which is something that can happen when you're aligning to a reference genome. You have to make sure you have at least absolute minimum of 4 gigabytes of memory, probably 8 is recommended on your computer. So I've restarted here, and I've also changed a couple of these settings at the bottom. So in the novel section, I've raised the read cutoff so we get fewer results, and so things run a bit faster, and the same with the score cutoff. I've also added an option here called strict and set it to true, and that tries to filter out some of the novel microRNAs that may be false positives. So this is something that can be switched on and off. So if we go back to, I'll just clear the screen, clear the console here, go to the command line, and we just run the command from there. It will launch, and what you notice is it tells us that this time it's going to map microRNAs, and it creates, rather than us having to, once we provided the species, it creates these mature and precursor sequences for the bovine sequences. And since I've already run this once, it's reusing the collapse files that we generated from the first run. And if you want to regenerate those for any reason, you can just delete the temporary folder that's in the that's inside the results folder, or just delete the entire results folder. So it's running. What it does is it aligns the reads to each library in turn, starting with the ones we specify here in the configuration file, and it'll then align to the mirror-base mature and precursor sequences, and finally it'll map to the align to the reference genome. And once that's done, it'll like last time produce a table of the fractions of each kind of each library that we've mapped. It'll, in this case now, because we're counting microRNAs, it'll try to count the isomers. It'll write them to a separate results file, and then if you provided a reference genome and have an index built for your reference genome, it'll try to do the novel microRNA prediction. So that's detailed in the documentation on the website, the GitHub page, which I'll give a link to at the end. But basically, this works by finding read clusters of aligned reads in the alignments into the reference genome, and it'll try to filter them if you've provided a cutoff for your numbers of reads in each cluster, and the rest it attempts to take the sequence, different sequences covering these clusters, and fold them into potential precursors that might be consistent with microRNAs, and predict the star sequence at the same time, the mature and star sequence. As you can see here, it's going to have to do this for 196 clusters. So we'll leave that running for a second. So once that's done, it will tell us that it's found 13 novel microRNAs. It's filtered them out, and then it's filtered some of the potential false positives out, and then it writes out the novel mature sequences and recounts them, remaps them to this by making a new index, which is a method that's used in other software as well, to create a final counts file for those mature predicted novel mature sequences. So if we go to the folder, the results folder, you see it's similar to before. We've a graph that shows the estimates of fractions mapped to each library. You can see here, always in this case, that the majority of the reads have been mapped to microRNAs, and most of the remainder are not mapped. Then it's also produced a CSV file for isomer counts for the novel microRNAs and for the mirror base. So just to show this example, these are the mirror base counts. So it produces for each microRNA a column of counts for each sample and normalized counts. If we go back and look at the results folder, the other file to discuss is the novel.html file. That's a HTML report created from the novel microRNA prediction. So if we double click on it and open it in a browser, I'll just zoom in a bit. And what that gives us is um, two sets of tables. And on the left side is a list of unique novel microRNA predictions. And each one has an ID. And for a lot of them, you'll see that they're mapped to multiple locations in the genome. So multiple identical precursors, which is commonly seen in real microRNAs. And these can be on the same or different chromosomes. So it'll show you the chromosome and the start location and the number of predicted mature reads. And each ID is a unique identifier, although it looks kind of illegible. It's actually a hash, so it uniquely matches or maps to a mature sequence. And the idea of this is to aid in replication. So if you run the software independently on two different studies or two different sets of samples, and you find the same novel microRNA, it'll give you back the same ID. So you'll be able to match between different independent runs of the software. And for each entry, there's a link internal link inside the page, and you click and it'll bring you to the entry for that particular microRNA. And each table here shows details of the precursor, the mature sequence, the predicted star sequence, the energy predicted for the structure, and there are two hyperlinks within each table. One is shows the the precursor coordinates in the genome, and links to the Ensemble browser for that region in the genome. So when you click on the link, it'll open a new page and show you where the sequence is located in the context of that position in the genome. And that's particularly helpful to tell you whether it's actually overlapping with other genes, for example. And you can also use Ensemble to view alignments, for genomic alignments. And then the other link is to the structure. So we use an external tool, an online web tool called Forna, to view the structures. And that may not be the greatest example. If we just go to the bottom and look at this one, look at the structure of this microRNA. This looks more like a regular microRNA hairpin, and the mature sequence is shown in green, the star in pink, and in this case the mature sequence is on the 3' side. So just to summarize, go back to this page. One thing to mention is you can see there's a score value in each table for each microRNA. That refers purely to the score for the precursor structure produced by the feature classification algorithm. And a program like MIRDeep2 has a more rigorous statistical model. So our system is more closely based on the mirror analyzer method for microRNA precursor prediction. And you can see that there are definitely improvements, a couple of improvements that could be made in terms of filtering and optimization for speed. And you should definitely, if you've used this program, try to use at least one other piece of software to compare the results to. There's also another program called Mirror Reader that can do novel microRNA prediction without a reference genome. So there are other tools out there. One of the selling points of this one is hopefully that it's a bit accessible, a bit more accessible and easier to use than some of the others. But you should definitely try more than one program. So just to summarize here, this is the link to the documentation on the GitHub page. It's the link under the wiki here. And for people interested in the command line tool, some of these entries here relate to the Python API. But there's a page explaining the command line interface that covers most of what I talked about in the video, including the configuration file and a few other features that I didn't talk about. And then there's also a page on the how to install the software. And just to say about the installation, that this software is designed to be run on Linux, and the in the video, the operating system I've used is Linux. But even if you are a Windows user and you're not familiar with using Linux, it really is fairly straightforward now to install a program called VirtualBox, or another virtual machine tool that allows you to run Linux inside your Windows operating system. So I'd encourage people to try that. It really isn't that hard. So just let's say a quick word here about downloading reference genomes. This is the Ensemble FTP download page for various annotations, FASTA formats, and the actual, it looks a bit confusing when you look at it initially, but if you scroll down to the bottom of the page, each column there and each format is explained fairly clearly. And if you're downloading a reference genome, you're looking for the DNA sequence, a FASTA format. So if we go back up, for example, to say we're interested to keep it consistent for cows. It's not obviously in this list, so a quick way to do it is just to say show all 50 entries. Scroll down and we see that the cow sequence is here. So if we click on this link for the genome sequences, it brings us to a HTML page with the links to each individual file. There's a FASTA file for each chromosome. If we're interested in downloading the entire, so in the examples I've given for the previous example for the novel microRNAs, when we do the mapping to the reference genome, we're mapping to an index made up from a sequence file with all the chromosomes, which is this file here. So if you're interested in doing this, then this is the kind of file. This is the kind of file you need. And the same applies to the. Sorry, let's go back. The same applies to the UCSC interface. It's got a link for each species. If I go to cow, I'll see that there's links to multiple genome assemblies. In the case of cow, there are actually two. And this more recent UMD 3.1, if I click on full data set, it'll bring me to a similar kind of interface where the links to each file are explained. And if we go down to the links here, in this case, it's this one that we want here. These are gzipped, so you would have to unzip them, and then you would build a bow tie index before it can be used in our program, and before it can be used for short read alignment. And the only other thing to mention is that you have to make sure that your genome sequence, if you're trying to say compare it to another study, you have to make sure you're using the same genome assembly as somebody else's, because coordinates can change between assemblies.